development of technology, to advance the technology, to come up with new methods, and so on and so forth. Very recently, I could read in one of the newspapers, and you are the experts to say whether it is a fact or not. Earlier, even a RT-PCR result, they were taking about more than 12 hours to get the results. And one of the laboratories has come up with within 40 minutes, results can be obtained. You have to assess the fact. I'm not sure, but you people are the authority to say that yes, it can be done. So see that even the development of vaccine, how many microbiologists and biochemist people must be working behind this particular development? Huge. And very glad to know that some of the students from this particular university are have been very actively working in this particular field. So I'm proud of you all. Teachers are producing these experts. They are training our students so that they can work at the national and international level, and they have been working. And that is how the things are going on. I'm very happy to see that Professor Abdullah Shohel, who has been associated with Shivaji University through Department of Microbiology and Department of Biochemistry since last couple of years. We have formally entered into MOU. But you know, many times MOU have been signed, but it takes a lot and takes a lot of time to really have exchanges, academic exchanges. I think this is one of the MOUs where the things were started first and then MOU was signed. I recall, I think one of his students also was here to get training in our laboratory. So student exchange also took part. I think he also visited Shivaji University some time back, and the things are going on. So I appreciate this particular collaboration and association between these two universities, and I must congratulate the faculty of life science departments for this particular collaboration. I learned from introductory talk by Professor Sonawane and Professor Zado that some of the speakers are our own students. And that is a great moment of pleasure. When students contributes and try to share his or her knowledge with the ongoing students, I think our students' confidence go up because they can see themselves in some time in future, they will again address this particular kind of gathering through their expertise. So it is a kind of confidence building process, I would say. This particular series of lecture has got so many such direct and indirect outcomes. But at the end of this particular series of lectures, I'm sure that students will gain a lot of insight in various domains, various subjects, various techniques, and they will have a kind of transformation and enhancement, the knowledge they would like to have in this particular domain. Thanks to the officials from Maharashtra Academy of Sciences, MASc, for supporting this particular activity. And of course, thanks goes to UGC for supporting such programs under STAT scheme. So such capacity building programs for the buddy students, young students, will go a long way and will be definitely helpful in all sense. I'm sure that in the next couple of days, you will have a feast of knowledge, though in a virtual mode. And I'm sure you will definitely enjoy all the lectures in this particular series. As a first in the series, Professor Shohel will be delivering his, I would say, the inaugural talk. This is, in true sense, inaugural academic lecture. And I must appreciate his presence. Yesterday, we had some different situation and we had to postpone this program. I must grateful and I must thankful to Professor Shohel to understand the situation and adjusting his schedule for today's talk and making himself available for this particular talk. I once again congratulate Professor Jadhav, Professor Sonone, and the team of academic team, faculty team of all these two departments for organizing this wonderful series of lectures. And my best wishes to the entire series and for such various academic activities in future as well. Best wishes. Have a great time in the next 10 days. Thank you. Thank you.
Thank you, sir. Now I request Dr. Mrs. P.B. Dange Madam for vote of thanks of today's inaugural function.
Hello, may I audible to you? Hello.
Yes, ma'am. Yes, you are audible.
Okay, okay.
Go ahead.
Thank you, Sneha. And here I am presenting the vote of thanks for first session. It's a good afternoon, all of you. It's a great and immense pleasure for me to have a president for this particular lecture series. Honorable Professor D.T. Sirke sir, dynamic personality, and as said, Dr. Sonone and Dr. J.P. Jadhav madam said that there is no any explanation or any words to explain what is or how much dynamic personality that is the Sirke sir has, and we are all experienced to that. Thank you so much, sir, for being a president for this lecture series. Secondly, I am also thankful for the honorable guest, today's guest, that is the Sohail sir, being for guest as a for this lecture series. Then all other honorable chief guests present, excellence teachers, and my dear friends, I am very much thank you for you, and I am thank university for giving me an opportunity to express my gratitude on this special day. It's a biggest day today. We have hosted the biggest online lecture series on this advance in life science research under the UC Stride component and Maharashtra Academy of Science Kolhapur chapter. In this series, we have invited eminent speakers, particularly from the biotechnology as well as the biochemistry department from the over the sea, and who will definitely helpful for our budding students as well. I'm also thankful to them. On the behalf of the Biochemistry Department and Microbiology Department, I express my heartfelt gratitude to our again honorable chief guest from his busy schedule, Vice Chancellor D.T. Sirke sir, being with us. Also, I'm thankful for this Abdullah Sohail sir from Jahangir University, Bangladesh. Pro Vice Chancellor Pro VC sir, Professor P.S. Patil sir, our registrar Dr. V.N. Shinde sir. I'm also thankful to Professor J.P. Jadhav, HOD, Department of Biochemistry, as well as the Sonone sir, Department of Biochemistry, and recently, as sir said, in charge of the Chemistry Department, Professor K.D. Sonen sir. I would like to thank all respected representatives who have placed for this particular lecture series and their presence also. Finally, I would like to thank organizing team, teachers and staff members. And lastly, I would thank for all the persons who directly and indirectly help for this program to run very smoothly. Once again, I am thankful to one and all. Thank you very much for giving me a chance to have a interaction with you. Thank you. Thank you, Sneha.
Thank you so much, ma'am. Now here I declare that today's inaugural function is over. Now I request.
Thank you so much.
Yeah. Okay, sir.
Thank you, sir. Thank you, Honorable VC sir. Thank you for your time. Okay. Good day. All right. So just now, inaugural function is over. I am again thankful to all the dignitaries who have joined this particular inaugural session. And now I request Professor Sohail sir to start his talk. First inaugural talk by Professor Sohail from Jahangirnagar University, Dhaka, Bangladesh. Professor Sohail sir.
So, am I audible? And how about my slides? Is it visible?
Yes, sir, it's visible.
Yes, sir. Yes, sir. It is visible and you are audible.
Okay. Okay. Thank you so much.
Supply of luminous on kVA. Inverter. That's what inverter circuit transformer. Yes, story inverter. Story inverter. 5000.
It's showing my slides are overlapping, so I have to maybe.
Slides are not visible right now.
Yeah,
Earlier
yeah.
it was.
Because message was showing my slides are overlapping.
Because earlier slides were visible, but right now they are not. Now it is visible, sir.
Okay.
Okay, please go ahead.
Good afternoon, dear participants from Shivaji University, Kolhapur, under the STRIDE program in this Advances in Life Science Research Series, ALSR-2022. Department of Biochemistry, Department of Microbiology, Shivaji University, Kolhapur, in association with Maharashtra Science Academy, Kolhapur chapter, Honorable President, Organizing Committee, students, faculties, and other members. I welcome all of you in this series, and I am again thankful to you inviting me in this very precious occasion. So during this COVID time, I think it's really a great initiative to arranging this lecture series. It will continue around 10 days, and I can find in the table program that lots of eminent speakers will be speak in each day of this program. And I came to know that among them, many of the ex-students of the departments. So it really brings that the productivity of the Department of Microbiology, Biochemistry, and others. I cannot repeat again because in my experience, I found this Department of Microbiology, Department of Biochemistry, and other department I was visited. It's really, really a wonderful job they're doing. Their research environment, expert faculties, and everything just awesome. And then the students and researchers are really working very hard to achieving their goal. So our MOU, I will show again about what we have achieved from the Department of Microbiology, Biochemistry. Unfortunately, we were planning to start collaboration with Jadhav ma'am because Jadhav ma'am was a very wonderful researcher and she has tremendous research outcomes. But due to the COVID situation, we could not continue. But I hope in future we will continue with Jadhav ma'am and also DS Patil sir and others. I hope when situation will in favor of us. So today I'd like to highlight few slides about plants and microbes in applied research to improve food and human health. It will not show the whole picture, but glimpse of something. And I would not speak about any details, but some highlights that will give you some ideas to think. First of all, I would like to introduce my university. That's Jahangirnagar University. If you visit here, Dr. K.D. Sonone sir once visited here, but I think he was in very short time. So we could not visit all the campus, but it's similar campus with Kolhapur University. We have lake, we have some upland, lowland, and lots of greenery around the campus. So it's a quite old university. I mean, the four largest university in Bangladesh. So among the four, Jahangirnagar University, one of them. And then total academic staffs is 755 plus, students 18,500. It's a rural area having 697 acres of land. And then it's nearby Dhaka city. From the international airport, it's only 30 kilometers, and it is from the heart of the capital, Dhaka, it's only 35 kilometers away. So here you can see few slides is showing is different parts of our university. And on most important things, during the winter time, lots of migratory birds used to come here every day, every year. And right now it's the peak time for migratory birds, and they are now in the full of our campus, so that every day many people during this COVID restrictions, lots of people every day used to come and visit this university to visit these migratory birds and others. So this is some slides, and you can see more in our university website to learn about the university and others. So I'd like to start with a few words about what biologists are doing. Actually, biologists means all of us, microbiologist, biochemist, botanist, biotechnologist, all disciplines we are working with biology. So we are working hard ensuring food safety, food security, disease prevention, treatment, green technology development, bioenergy production, developing genetically modified organisms, and tracking the role of microbes and plants in climate change. So this is our basic roles. That's why we are doing research, we are teaching, we are developing our researchers and students and others. So these are the major goal we are working on. So about the microbiology, from everywhere, the existence of microbiology from food, feed, plant, soil, everywhere these microbes are existed. So sometimes they are good, sometimes they are bad. So not always they are bad, but they are acting very good stuffs every moment we are working on. Our microbial planet, so thinks microbes are bad guy, but think again that every time, are those bad? Really are those bad? So microbial planet, from the bottom of our land to the air, so everywhere their existence, and they are doing their contribution a lot from our guts to the environment. Everywhere we can see their contribution. So key topics in microbiology, what aspects are now. Number one, this antimicrobial resistance, environmental microbiology, pathogenicity and virulence, and then biotechnology and synthetic biology, then covering microbiomes, and then food biotechnology. So that's not all, but the few that microbiology right now contributing. Just one example, microbial decomposition of organic matter. All of us, we are very much known about that, how microbiology helping decomposing the dead bodies or when these dead bodies are decomposing and our environment become clean, and then different nutrient and other cycles are there continuing to produce or make this earth or make this planet is lively, green, and energetic. So this is the microbial decomposition that are helping us to sustain this environment. Human microbiomes, plant microbiomes, and marine microbiomes. If you think about that, how much their contribution and how much they are doing, so we will be really, really amused that microbiology is not our enemies. So microbes are always our friends, and they are so much friends, and then how they are contributing to save our lives and how they are contributing to ensuring the better future, better environment. Microorganisms and climate change in marine and terrestrial biomes. If you have a look that naturally or this basically microorganisms, they are helping to clean the environment. But as a human being, what we are doing? So all sorts of climate change issue, the reasons behind that climate change, adverse climate change, humankind, and our different devastating activities is making these climate change issues. And those we are destroying the environment, we are destroying the microbiomes, we are destroying the soil, destroying the water, destroying everything, and then the climate change, adverse climate change issues are happening. So due to our act, we cannot say for others they are involving in that issue. Totally, solely, we are acting on that. So agriculture and other human activities also that affecting the microorganism. We are now industrial revolution happening. So everywhere we are building, we are establishing industries, but very few we are getting attention to make the safe exit of wastes to the natural environment or fertilizer or other stuffs, or we are producing the different types of cropping system, but we are destroying the environment. Microbial diversity we are destroying. We are so much depending on these chemical fertilizers than others, biofertilizers and everything. So these are our acts that really, really affecting the microorganisms, and in long run, they are also affecting the agriculture and the human, threatening their future in a very, very alarming way. Climate change, what they are doing at the end results, I mean, not I'd like to say the end results, the resulting fact of the climate change that exacerbates the impact of different sorts of pathogens. I mean, how COVID-19 arises, we know that this coronavirus pandemic, and we are facing the effects. So different types of disease that like black famine and other disease, different types of zoonotic diseases, different types of lots of climate stresses, also marine life. So if we see the terrestrial, aquatic, marine, everywhere we are threatened by different types of microbes. And then someone, because no natural control between them, so that someone is very much getting uprisers, and then they are doing their devastating acts to us. What we need to do, we need a clean planet. So we have to think about ecology. We have to innovate all green technologies to reduce carbon emission. We have to think green, go green, live green, and also act green for the sustainable green planet. If we can do as a biologist, microbiologist, biochemist, botanist, so if it's our goal or target, then definitely we can make a green planet, sustainable green planet for peaceful and very lively living in future. We have to think that. We should think the act positively to our planet to protect our biodiversity, but not by hypothetically, because all government, all person everywhere in the world, we are hypothetically all we want to save the world, but our acts is not supporting our hypothesis. So protect the biodiversity, not by hypothetically, but act or do practically. Now, microbiological threat. So what are the microbiological threats? Right now, all we know that this different kind of infectious disease are global hazard that puts every nation and every person at risk in everywhere. Coronavirus outbreak is a prime example, and neither geographic nor political borders can prevented this COVID pandemic or movement of this COVID-19 or this coronavirus. And then they arrived silently and lethally. Microbial pathogens constitute a grave threat to the health of humans. Throughout the history, humans have struggled to control both the causes and consequences of infectious disease, HIV, different types of flus, Ebola, plague, and lots of disease from the beginning of the human history. So it's become so familiar, and then their effects, all of we know, those though all are history, but we know. I'd like to focus few things about the invisible threat to public health, the extended spectrum beta lactamases producing E. coli bacteria. So this work we have done this last 2 years. E. coli and their changing, their formation, ESBL formation E. coli in fecal sludge to the Rohingya camp in Bangladesh. So what is this fecal sludge? A mixture of human excreta, water and solid waste. Fecal sludge is one of the major source of E. coli, and that can produce extended spectrum beta lactamases. Fecal sludge also contains many pathogenic microorganisms, and inadequate personal and domestic hygiene also play a role in spreading diseases. So everywhere, every community and every human beings where they are living, so they have the fecal sludges. Some we are so optimistic, so we are treating them, but most of them they are going to the environment without any treatment. So what is going on and what's happening? So over uses of different sorts of antibiotics, different types of drugs, different types of adverse effects. So those E. coli in the these fecal sludges, those E. coli, they are living there. So they are become extended spectrum beta lactamases. They are producing there. So they are not working against the third generation, fourth generation antibiotics as well. So our future is under threat really due to the antibiotic resistance. So our articles was published in Frontiers in Public Health in last year. So if you are interested, you can have a look about that. So antibiotic resistance is one of the major threats to global public health, and WHO enlisted it as one of the top 10 threats in 2019. Excessive use or misuses of broad range antibiotics in medicine and agriculture is the most prominent reason for the development of antibiotic resistance. I am not focusing here about the COVID-19 or coronavirus and the pandemic, because this COVID threat, this in front of us, media coverage is huge media coverage, and all of us every day we know how much persons are affected, how much death, how much this national and international economy are losing and everything. But how about those invisible threats? is more people are suffering, more people are dying every day due to this AMR, antimicrobial resistance facts, but no one is caring about that. So that this is the silent killer. We are focusing about something that are visible, but we are not focusing that are more threatened but invisible. So we have to think about that. We have to think. So antimicrobial resistance, all we know that bacteria can attain antimicrobial resistance through many mechanisms, including enzymatic inactivation of antibiotics, target modification, and dynamic efflux. Our study was aimed to investigate the prevalence and molecular characterization of ESBL producing E. coli in fecal slush. Samples collected from fecal slush treatment plants in Rohingya camps, Bangladesh. So, because we know that all of you know this Rohingya, these millions of Rohingyas, they are living in camps, and from where they are very densely populated. Though different international initiatives, they are trying for their better living. They are different waste they are treating, but after the treatment even, so due to the large population and large amount of waste generating every day, so we choose these areas and then we found the very disappointing information that these lots of ESBL producing E. coli are producing even after the treating the fecal slushes as well. So that means the treatment was not properly going on. So we have to focus on that. So I would like to highlight some results, not all details, because I already told that I am not boring you describing some details, results, everything. I would like to highlight some few outside things. That's why you can think for future endeavors. So this is the fecal slush treatment plants in the Rohingya camp, where slushes are coming and then going to the environment after the basic treatment facilities. So we have collected the samples from those treatment plant and bring in the Dhaka ICDDRB laboratory, and then we have identified those microorganisms, and they are so what we have found, the results, if I move to the final results. So this is the alarming results we got. Total 27 isolates we have found, this ESBL producing E. coli. All of those were resistant to ampicillin and cefatoxime. 93% of the isolates were found to be resistant to the cefuroxime. Cefuroxime is the third generation and the most widely used antibiotics right now. 26% isolates were found intermediate to imipenem. So if this is the issues, so that how is happening, this antibiotic resistant patterns, this ESBL producing pathogenic E. coli. I mean, this fourth generation antibiotics even sometimes is not working against them. So that similar cases was happening in the hospital waste or other household waste and everywhere. So ESBL producing bacteria, their numbers, their prevalences are increasing day by day. So we have to be very careful on that, and our act should be how to control them and how to control their activities. Now, I'd like to focus few slides about this plant based research in Jahangirnagar University. Due to the short period of time, I could not address all of those, but few I will focus here. About plants, this is very wonderful slides by Daniel TAXS. So this plant, and within this infograph, it's very clearly mentioned that in the plant and different parts and their activities, roots, stem, bark, leaf, fruits, and photosynthesis, oxygen, and inner and outer parts, how they are acting and what they are doing about the plants. So we cannot say much more about the plants because plants, we know the what plants are doing for us in everyday life. The benefits of plants, this is from Urban Planet website. I took these slides from Urban Planet website. We know what plants are benefiting us. They are making the harmony. I mean, they are uptaking carbon dioxide and they are releasing oxygen, and we are oppositing, we are releasing carbon dioxide and we are uptaking the oxygen. So between plant, animal and plant, so they are making the harmony between us in the environment. Air quality, we know that dust, bacteria, molds, and others, and even plants, how they are acting. One plant for every day, 3 people improves air quality. So health, say minor illness and different types of illness. So plants, how they are helping them. We can see this. I mean, Jadhav madam is working on medicinal plants, I know, and then she is doing very great work about the inside. I mean, how the secondary metabolites working to defeat different types of disease, and he's also every day in his research work, he's finding the potential secondary metabolites, their functions and everything. So all of we know that the health issues, how much the medicinal plants they are contributing. Productivity, we know the productivity, because all crops we are depending on food, feed and everything. So the productivity depending on the plant productivity, and plant is getting the energy from the sun, and then through their production, I mean leaf, root, beans, seeds. So we are getting the energy. So they are also supplying the energy to us. So, but a few problems on this plant-based medicine production. I am focusing on only this plant-based medicine, not others in this lecture. So what are those problems? Cultivation problems, manufacturing problems, marketing and distribution problems. Cultivation problems means lots of plants in the nature, like in India, Himalayan region, and many others, Western Ghat and others. So lots of medicinal plants are existed there. And then local tribal people, since the history of the human, I mean, civilization, they are using different parts of the medicinal plants to healing different disease and disorders. So right now, there are increasing demand of the medicinal plants, and then worldwide demands, export, import, and other facilities, so that so many plants are not yet well botanical characterized, so that adulteration with wrong plant species is a very common act right now is doing. Field production, I mean, those plants, they are growing in the natural environment, always threatened to the fungi, bacteria, insects, weeds. So environmental pollution, lots of heavy metals, organics, herbicides, and pesticides also affecting their growth and development. Crop nutrition, altered profiles of medicinal metabolites when they are culturing in the different environment that it's original. So that the several cultivation problems right now facing for the different medicinal plants. Then during the manufacturing steps, harvest, drying, storage, processing activities are losing day by day. I mean, due to the lingering the steps of harvest, dry, storage, processing. Finally, marketing and distribution, this advertisement is exaggerate of efficacy or falsified the efficacy, false efficacy of a certain medicinal plants, they are also in the major problem for marketing and distribution. And after that, so that these few steps we have to address, I mean, in biotechnological approach or laboratory approach, we have to enhance to overcome these three different steps. So what will be the answer to overcome those aspects? I mean, not fully, but partially we can overcome. So in vitro culture techniques for the production of active compounds in different plants is giving some results of those problems already created. So cell cultures, organ cultures, or whole plant cultures can be done in a laboratory scale or commercial industrial scale that can produce 2 to copy plants, the same similar organs, similar cells that are producing the novel phytochemicals or novel chemicals. So these slides several times I have shown in Kolhapur University. Nature to the bioreactor. I mean, bioreactor is a device in where we can large scale produce the plant cells. I mean, valuable plant cells we can produce. First of all, we can introduce the culture in the culture dish, and then we can choose the cell lines, and then we can go for small scale suspension culture in conical flask, and then we can move forward to the laboratory scale bioreactor, and finally these laboratory scale bioreactor can be much more commercialization through industrial scale bioreactor. So the workflows, I mean, this medicinal plant research, what we can do, we can select plant, then development of cell culture, flask culture, bioreactor culture, we can collect fresh sample, and we can do antioxidant enzyme, secondary compound analysis. But after harvesting or collecting the samples, we can make it dried powder, and then we can make the extracts for phytochemical analysis, antimicrobial analysis, anti-diabetic, anti-helminthic, free radical scavenging activities. We can characterize other antimicrobial assay. We can do cytotoxicity assay, UV visible spectroscopy, ACM, FTIR, XRD, mass spectroscopy. We can do lots of things. And then we can also tested the efficacy in the cancer cell line, non-cancer cell line. So this is few workflow that we can easily do in a mid-range of plant biotechnology, biochemistry, microbiology laboratory. For this, we no need very sophisticated one, but sophisticated laboratory. But I believe in the Kolhapur University, you have very sophisticated facility in Jadavsman lab, and also DS Patil sir lab and others lab, Sonone sir's lab. So all of these facilities you have, but these all facilities in Jahangirnagar University till now we don't have the full facilities, so that we are collaborating with you. We have the basic facilities and you have the advanced facilities, so we can work together to make answer, I mean the great answer on that. Another part of work, this in silico part, that you have very nice facilities in Sonone sir's laboratories. This ligand preparation, target identification, protein preparation, energy minimization, receptor grid generation, molecular docking, pharmacokinetic studies on Alzheimer's, Parkinson's, neurodegenerative disease, Zika virus, Ebola, HIV, and others. I mean, we have to target with active phytochemicals, which one will be the effective one. Then we have to choose target and research direction, and then we can work on in silico part to establish our hypothesis or prove the hypothesis what we have done. Another very simple work we did before the COVID pandemic, that health effects of street vending aloe vera juice and leaves. Why we did that experiment? Because we know the aloe vera and versatile uses of aloe veras, but I am not sure about the Maharashtra and Kolhapur, but in Bangladesh, this aloe vera juice is very popular, and the street vending aloe vera juice is quite available in everywhere around the campus, outside the campus, in the streets, markets, and everywhere. So we are taking those juice, aloe vera juice, for various health benefits on aloe vera. But actually what's happening? Are we getting the benefits, or we are getting different kinds of disease threats and others? So we did those experiments, I mean, to study the prevalence of the disease, the contamination on others, transmission, disease transmission, so that vending site, we check the vending site, water utility, water source, personal hygiene, adequate protection and covering, lacking in cleanliness and improper storage. So we have studied the following things. And unfortunately, we found lots of bacteria, pathogenic bacteria in those, but not in the aloe vera, natural aloe vera, but those from the aloe vera juice. They are coming from different sources. They are for preparation of aloe vera juices. So we recommend that these vendors should be followed 10 or 15 rules to avoid those contamination. And when we are taking, we are also recommending for the customers as well to check those precautions before they are getting sick. So plant antimicrobial studies, that you know that it's very nice field for antimicrobial studies, medicinal plants, make it extraction, purification, then identified the compounds. Finally, we did the antimicrobial studies and which extracts working on, to preventing which microorganisms. So it will be a different research dimension that is helping to identify the antimicrobial research, and then it's very quite common in every labs they are working and getting the tremendous benefits as well. So we have tried this big jar Orellana plant, like commonly known as Asoite lipstick tree. I think Maharashtra also you have lots of these plants. So we have studied in fruits, bark, stem, seed coat, and others. And this works also published in a good journal, how much antibacterial potential also have this big Orellana plant. And then we are very much fond of different types of spices. This is cooking ingredients, everyday cooking ingredients. We are using these 10 common spices. And then we have checked, how much antimicrobial and other effects those spices individually they are they are contributing on us. And then we found that this very good number of antibacterial activities on those regular spices we are taking. So it's getting our everyday life is easier and protective. That's why, not that's why, because traditionally we are taking, but ultimately that's giving us different types of prevention through our this regular food habit or food practice. Now, I'd like to focus about the collaboration between Shivaji University and Jahangirnagar University. That these slides I already explained in front of Vice Chancellor Sir. Our collaboration started before MOU have signed with Dr. K.D. Sonoane sir in 2018 when I was visited Maharashtra, Kolhapur University for attending a conference, I think in different parts of Kolhapur, and then I visited Kolhapur University, and I found that this research environment and research initiative enthusiasm is really, really wonderful. And then I talked with Sonoane sir how we can start our collaboration and how we can go for. And then we started different types of research work in 2018. Finally, we proceed for MOU signing in 2019. Between the work between 2018 and 19, we have published 2 research articles in 2020, I mean 2019 and 20 in a good journals, and 2 other work. That work have done by VC sir mentioned that one of my students already visited there, and then he got a training from SARS lab, and then he did really, really nice works in his MS thesis, and then those publications under progress, and we believe that we will get very wonderful publication, and we are expecting those publications will be impact factor not less than 5. We are expecting more, but at least 5 impact factor journal we can publish at least 1 paper from those studies. K.D. Sonoane sir was visited once in Jahangirnagar University, and officially I also visited once. Though I am maybe 3 times I have visited, but officially I visited once. A training and workshop during the COVID pandemic. One training workshop was conducted, and Honorable Gaurav sir was the trainer for the leadership training that we have conducted during the COVID pandemic period. I mean, during the last 2 years, I mean, maybe last years. And then we have attended e-conference 4. The SAR also attended here. I also joined over there. So we have around 4 e-conferences. And then 1 student from Jahangirnagar University, Siam Ahmed, visited, trained, I mean, I will not say visited, because Kolhapur University was giving us a chance for a student exchange, and then he was there and he trained, and it's really asset for his research career. Here you can see a few slides. This is my student Siam Ahmed, and this is we have during the MOU signing. At that time, Vice Chancellor sir and I visited his office, and our MOU was focused in the I mean, this is maybe Marathi newspaper, and then this is few visit slide with registered sir and also PRO, and this is the in front of your I mean this main building, and maybe this is a Ghule sir, I think. So we had very nice times while we are visiting there. So during my student visit, actually, we were working on Panax ginseng adventitious root culture in bioreactor in Bangladesh, and in silico parts anti-Alzheimer's activity and others active pharmacological agents and activities. Siam Ahmed was studied in the SARS lab. So here I would like to say this is ginsenoside components we worked on. I mean total 8 components, RB1, RB2, RC, RD, RE, RF, RG1 and compound K. And then I'll focus with slides. And then molecular docking of ginsenoside against Alzheimer's, amyloid beta protofibril, what we did in the SARS lab, and the bind docking of compound K with the amyloid beta protofibril, and then the results. These results I told that we are planning to have much more. We have some others, docking and others molecular dynamics works added with this work, and these results now under preparation for publications. And both, I think, Shonwana sir, we will send the manuscript draft very soon for quick review, and we can submit. So that's almost end of my lecture. Now the conclusion: why are the products of cell suspension culture different? Here, this is the original root of ginseng root, and this is the cultivated root in the in vitro systems. So this is an example, but in the most cases, the similar ways. When we do culture in vitro, it's quite different than the original one. Though metabolites profiling was not different, but its physical appearance is different, because lack of differentiation and organization within the short space, because we are cultivating, we are culturing in a vessel that is closed vessel, so they don't have much space to differentiate it. Necessary for the expression of many plant secondary metabolites, so that we did lots of media composition, hormonal composition, and others associated nutrient composition optimization. So it would be the reason. Disrupt regular metabolites pathway and results in the accumulation process and the desired compound, so that sometimes the difference could happen with the original ones. In the large scale production, rather than the natural production, few challenges also need to overcome. What are those? Cell culture establishment is the first challenge, because not all plant species are responsive to the tissue culture. So if those valuable medicinal plants are not responsive to the tissue culture, then we cannot proceed further. And then even though they can respond for cell culture or tissue culture or callus culture, but it's very difficult to get stable cell line. Even though we get the stable cell line, the production is not even. Sometimes one batch is producing very well, another batch is getting worse. Reduction of biomass, and then chances of contamination. So in every steps, we have to optimize. After optimization in each steps, we cannot proceed for further. Optimization in solid culture, optimization in small scale liquid culture, optimization in the small scale bioreactor. Then we can move forward for the final or a larger scale bioreactor, because we cannot do optimization in the 10 ton, 15 ton or 1 ton bioreactor. We can do optimization in 2 liter, 3 liter, 5 liters bioreactor, but we cannot do up. So we have to optimize all our experiments. Even though we optimize in the solid culture, we must similar optimization we have to make in the small scale liquid culture. Similar optimization we need to do in the small scale bioreactor culture. So it's also time consuming, and we have to focus on that. High initial installation cost, need for training trained technicians to overcome the reduction of biomass, contamination challenges, and other production issues. And then that already challenges can be overcome through tissue culture optimization, the elite genotype selection, and frequently development of new cell lines. I mean, we cannot use one cell line for a long time. So every year we have to develop new cell line, and we have to follow the good culture practices. And then metabolites, how do we enhance the metabolites? Because from the natural to the in vitro culture, metabolites, initial metabolism, though the profile of the metabolism was the similar, but the quantity of metabolism is not similar. So we have to enhance the metabolites content. How we can do that? we can do gamma radiation, we can do transgenic genome editor plants, we can do chemical elicitation. So most of the case, we can enhance the metabolite contents using different types of chemical elicitation or biological different sorts of elicitation. And a chemical elicitation can improve the 2, 3, 4, 5 times enhancement of the secondary metabolite. So we have the answer to enhance the secondary metabolites. So plant-based medicines are becoming increasingly important to the world health and economy. And then biomass production with control the quality of plant material and time of delivery. We should focus on that. Independence from environmental, seasonal, geographical, and political constraints, so that we can do we can produce any plants anywhere in the world, control growth and development to maximize the bioactive contents, consistency, quality, and yield of phytochemicals. We can enhance biochemical characterizations of active ingredients. We can do, and we can enhance the facilities. So that's all for today. And we have an initiative called Science Portal Bangladesh. Science Portal Bangladesh is a science communication initiative. We engaged young people to communicate science for all kinds of people, positive science. So if you are interested, you can visit. We have Science Portal BD web page and also Facebook page. So you can support our initiative and you can help to enhance our initiative. So I'd like to acknowledge Jahangirnagar University, Department of Biotechnology, Ministry of Science and Technology, Ministry of Education, Bangladesh Medical Research Council, Cell Genetics Lab, UGC, and Bangladesh Livestock Research Institute for providing us funds for the different projects ongoing and accomplished project. Thank you so much. Thanks again for giving me a great opportunity to speak in front of you. I am not a very famous researcher like Jadhav Madam, Sonoane sir, or something, some of your esteemed professors, but I am trying my level best to achieve something with the help of you guys. And then I do appreciate further cooperation and collaboration with Jahangirnagar University Biotechnology Lab, with the Shivaji University Kolhapur in different departments and under the leadership of K.D. Sonoane sir. Thank you so much. Thank you for your patience here.
Thank you so much, sir, for such an informative talk. Now I request Dr. P.K. Pawar sir for giving vote of thanks.
Thank you, Sneha. Respected Chairman of the session, President, Honorable Vice Chancellor, Professor Shirke, then the organizer of this webinar, Professor K.D. Sonoane, Head of the Department, Professor Jadhav, our senior faculty member, Dr. Dandge, and today's guest of honor, Professor Sohail from Jahangirnagar University, Bangladesh. I have been assigned to begin the end of this wonderful session. This is wonderful, beautiful. So what to say? Such an informative talk, very well illustrated. We are very happy to have you, sir, as a inaugural speaker for today's webinar. Now, dear participants, this is the class example of what kind of lecture should be. Very informative, very illustrative, and maximum use of figures, and whatever he is doing in his laboratory, that is presented holistically. That is beautiful. So what to take home? The take home message from today's lecture, what I could understand, he covered two topics: microbial as well as plants' role in human life. And he had addressed a very burning topic which is discussed worldwide, the problem of these Rohingya camps. And he has addressed the pollution that is associated with that. Millions of people, those are living in the camps. and he had dared to take that challenge. And it is kind of, I would say, a class example of the research which can be regarded as the lab to land research. It is starting at lab, processing is there, and then he is recommending something, that what is to be done for treating that environmental pollution problem. So that is beautiful. And today, the antibiotic resistance or antimicrobial resistance is a really burning threat. He mentioned that WHO also have identified top 10 threats, and one of them is antimicrobial resistance. I agree, and everybody should agree. Every life scientist, including the students, they should agree with him, and we agree with you, Professor Soel. This is really a burning question, and he had proposed some solutions also. Identified 27 strains with multiple spectrum of the resistance, and he had identified some superbug kind of things. I guess 7% out of 7 strains out of 27, they are most resistant. Okay, so this is one part, and second part was the offering the solution for the drug requirement, plant-based drug requirement, using the technology, using the biotechnological tool that is called as plant tissue culture, the in vitro cell culture. This is one of the promising solutions for the requirement in these challenging times, whatever we are facing. I'm not exclusively focusing on COVID-19, but there are many microbes who have a capacity to infect. And in my opinion, in my personal opinion, because plant tissue culture is very close to me also, because my topic of PhD was also plant tissue culture. So I am 100% in agreement with Professor Soel that yes, the promising solution or promising source for the modern day drugs could be the plants. But those cannot be extracted directly from nature. Those can be cultivated in laboratory in controlled conditions, and the continuous source of the pharmaceutically active compounds can be generated. But he discussed some challenges in that also, and tried to propose the solutions for those challenges in the workflow, what can be expected. Okay, so this was really a wonderful, really a informative lecture. So on behalf of Shivaji University, on behalf of the organizing committee and the host of this webinar, Professor Sonone, head of the department, Professor Zadav, and our faculty members, as well as from participants and the organizing committee, I wish to say thanks to you, Professor Soel, and along with that, I would like to assure you that in future, our MOU, whatever is there, that is going to be flourished in multi directions. So we offer all the help from our side, and definitely we are going to go way ahead in coming future. So all type of in silico, in vitro, animal-based studies, all the things, those can be done here. So we would be happy to share our expertise, and we would be happy to adopt your expertise from your side. Okay, so I assure from our side, this MOU would be a great success in coming future. Thank you very much, sir. Hope to see you soon. Thank you.
Thank you.
Over to you, Sneha.
Sneha.
Thank you so much, sir. Now I declare that yes, today's session is over.
All right. Okay, Professor Shoel sir, thank you very much for excellent talk. And thank you very much for accepting our invitation.
Thank you.
For the chief guest for the inaugural function.
Thank you for inviting me.
Yeah, thank you very much, sir. Thank you very much, sir.
Thank you.
Okay.
Thank you.
Good day ahead.
Okay. Yes.
Okay, good.
Thank you.
Okay, thank you.
Thank you, sir.